evening, everyone. My name is Professor Tara Dean, and I'm the Provost for London South Bank University. It is my absolute pleasure to welcome you all to the inaugural lecture of Professor Eiman Aleem. In particular, I want to welcome the students who have decided to come to the lecture this evening. Really appreciate you being here in this autumnal evening, listening to somebody who probably has inspired you and has taught you in your time with us. So the presentation of inaugural lecture is a significant milestone in the academic career of a full professor. It provides them individually with an opportunity to present their career so far, to introduce their field of expertise with a wider audience, and to update colleagues in current and their future plans. For the schools where our professors are based at, the event is a chance to celebrate the achievement across the staff and student body, to strengthen existing relations and catch up with alums, if there are any alumni here, and also to establish new conversation and collaborations. For the university, inaugural lectures represents an essential component of the public events program. It is an opportunity for us to engage with general public, creating a wider awareness of universities' contribution in teaching and research. So as such, we are delighted to be restarting this again for the academic year 22-23. We record all our inaugural lectures, and they will be available on our LSBU YouTube channel, and this lecture will be there in a couple of weeks' time. Of course, meeting in person for these lectures is very special. There was a period that we couldn't meet in person, but meeting in person really facilitates a greater richness of understanding and also really offers flexibility in the post-event discussion, hopefully informally, to talk to Eiman a little bit more about her work and her career to date. Of course, meeting in person also has the opportunity for me to rehearse that old age tradition of any face-to-face event, so please bear with me. Switch off your mobile phones. Keep in mind that we are recording this event. The toilets are somewhere signposted outside. We're not expecting any fire alarms, so if you hear the fire alarm, there is a sign at the back of lecture theater and here, please exit in an orderly fashion. So, allow me to introduce Professor Aleem. Eiman truly exemplifies the saying that science has no geographical boundaries. Eiman grew up in Egypt and completed her undergraduate and master's studies at Alexandria University in Egypt. She then moved to Germany, where she completed her PhD in molecular cancer biology and biochemistry at University of Heidelberg and the German Cancer Research Center. She then moved back to Egypt for a period and moved through the academic ranks before joining the renowned Karolinska Institute Cancer Center as a research associate professor, where she led research projects and supervised PhD students and research fellows. She then decided to move to US, where she held the associate professor role in the College of Medicine at University of Arizona, as well as a research scientist role at the Institute of Molecular Medicine at Phoenix Children's Hospital in Arizona. Again, in that setting, she led a number of projects in her field. In 2018, she saw the light, and she came to UK, and she joined the London South Bank University, initially as associate professor in biomedical science, and was given the responsibility to lead the design and development of biomedical science degree at London South Bank University, and also to secure external accreditation for the program. Both of these she has achieved with diligence, hard work, and commitment and collegiality. Incredible achievements for someone who prior to this had very little experience of working in a British higher education setting. In 2019, she became the fellow of the Institute of Biomedical Sciences, and in 2021, she was promoted to a full professor. Earlier this year, she became a senior fellow of the Higher Education Academy, demonstrating her commitment to high-quality teaching and her leadership of a key provision for the university. So in deciding what else can I say about her, I approached Steve Hunter, her head of division, and asked him to ask her colleagues how they will describe her. And I received rather extensive commentary, all very complimentary. But perhaps this quote from one of them sums it all. Professor Aleem is an exemplary role model to her team, consistently demonstrating a commitment to upholding the university's core values and encouraging others to do the same. She is a supportive leader who is committed to improving student experience. What a fantastic thing to say about any colleague. Beyond her school and students, she is an excellent academic citizen of the university, and I, for one, am particularly grateful for her leadership of the GenderNet, one of our staff networks. I also asked Steve to find out outside the work what makes Eiman rather interesting. And what I wasn't prepared for was this long list of hobbies that Eiman has. So let me tell you, and some of these I had also accompanied photos and images to go with this as a testament of her activities. So perhaps it won't surprise you to know that Eiman loves traveling. In fact, she has visited 39 countries and 322 cities and counting. She is a very keen photographer and is studying for an online course in professional photography at New York Institute for Photography. I hope this is not in her workload allocation model. So, she's a painter and particularly likes pastoral painting. She loves scuba diving, snorkeling, adventures such as paragliding, parasailing, desert safari. And these were just a handful of what I was sent to share with you. I must admit, after reading the list and seeing the photos, I realized that I lead a rather dull life. And I had a chat with Marc Anthony. We need to pick up our hobbies as leaders within the institution. So in sum, she is a great colleague and an inspiration to her discipline, to her students and staff. And we are very proud that she has chosen London South Bank University as her academic home. So please join me in welcoming Professor Aleem to deliver her inaugural lecture.
Good evening, everyone. I'm sorry the microphone is not working properly, so I have to stick here in this position. Thank you very much, Tara, for the wonderful introduction. And thank you, whoever wrote these nice words about me. Thank you very much. And I thank LSBU for giving me the chance to talk about my story, my journey, and my achievements in, or contributions rather, in the field of cancer research. So preparing for this lecture made me go back in time for 25 years, just reflecting on the last 25 years of my life. As you see here, I was still a PhD student at the German Cancer Research Institute in Heidelberg. I was like some of you students here. I'm, by the way, very thankful for the students that I see from year 1, 2, and 3 who are here now, and I hope it will not be boring to you. So yeah, at that time I was still learning the basic stuff in molecular biology and cancer biology, and then from there I had also a journey full of ups and downs. And then before I start talking about the most challenging part of this talk was that I know that the background of the audience is not in cancer research, so I had to make it as accessible as possible. So please, if you cannot follow for some reason, just let me, I'll stop and explain, okay? So, okay. So I'm gonna talk about the cancer problem, the magnitude of the cancer problem, my contributions to 3 research topics in the field of cancer biology, and a word about the future. So let me start by asking you this question. Who in the audience has not watched a loved one suffering from cancer, or have not heard of anyone who has cancer? Speaking of myself, I have many. Neighbors, friends, relatives. So unfortunately, it's a very serious disease that claims the lives of so many people. And to put a perspective in numbers, cancer is the second cause of death worldwide after cardiovascular disease. Some facts about cancer before we dive in the research topics. So there are 5 behavioral risks or dietary risks that are associated with death from cancer. These are high body mass index, lack of physical activity, low intake of vegetable and fruits, as well as tobacco and alcohol use. And when Lynn Dawkins is in the audience, I cannot talk about tobacco use. I'll talk about it from a different perspective. And thank you for coming. So tobacco use is the most important risk factor for cancer and is responsible for 22% of cancer death. There are also infections that, like hepatitis B and C, and Helicobacter pylori, if you have heard about it, it's a microbe in the stomach. Human papillomavirus, these are all responsible for about 25% of the cancer cases in low and middle income countries. And cancer is a very, very expensive disease. In England, I read that from the NHS, it costs about 5 billion a year, and for the society as a whole, because the cost also includes loss of productivity, could reach about 18 billion pounds on a yearly basis. So having said that, let us focus on some numbers regarding incidence and mortality. And these numbers are from the year 2020, covering all cancers, both sexes. What you can see down here is the number of cases, 19 million, suffering from cancer in 2019, And on this side, about 10 million died of cancer in 2020, not 2019. The estimated numbers for 2040, in 20 years from now almost, is 28 million or 29 million. The number of deaths is estimated to become 16 million in 20 years from now. So we got to do something about that. But the good news, if we can say that this good news is that, for example, breast cancer is number 1 in the incidence, but number 4 or 5 regarding death from cancer. The same thing for prostate cancer. It's number 4 in the incidence, but about number 8 in the death from cancer. This gives us the idea or the impression that these two types of cancer are becoming more and more like chronic diseases thanks to cancer researchers. And the earlier we detect cancer, definitely the better, the higher the chances to recover. Lung cancer remains number 1 killer regarding death from cancer. So where did cancer come from? You guess, you tell me. From Egypt. And this was the first recording of cancer was in the so-called from Edwin Smith papyrus from 3000 BC. And this papyrus describes cases of breast cancer and bone cancer. And although it was not used, the word cancer was not used, but the tumors were mentioned and how they removed it. And that also, it was written in this papyrus that cancer has no cure. So they were very good with medicine at that time. And the god of medicine, he's called Imhotep. He was the one who wrote the papyrus. Or taught me to do was always to document. So I have journaling. I journal from the age of 9 years old till now. So I can write a book, really. So what happened over the years, as Tara said, I didn't realize I've traveled that often, except when I reflected on all that, all the issues and all the research I had. So I studied biology in Alexandria University. I studied doing masters in cancer, by the way. The reason why I started working on cancer was when I was a teenager. I visited one of my aunts who was in terminal stage breast cancer. And that was the first person I've seen in my life dying. She died after 10 days of this visit. And she was screaming and crying loudly that I was terrified. But at that age, I didn't know that, someone could be in that much pain from a disease. And at that time, when I was still a teenager, I said, I got to do something to alleviate a little bit the suffering of cancer. I was still in school, not even university. And then I started, since I was in a master's, doing masters, I did continuously cancer, PhD, postdoc, and so on. So the general goal I had for my research was to better, understand the molecular biology of cancer in order to develop targeted therapies and better predict response to therapy. What is targeted therapy? I will go through those basic definitions before I talk about my research so that people can follow me. And if you don't follow me, please let me know. So my research in cancer biology falls under 3 topics. One of them related to a protein called Insulin-like growth factor receptor and how we can target this in cancer. And there are subheadings or kind of related projects under the main heading. And also understanding how the cell division cycle works and how can we target it in cancer. The third topic is omics, and I will explain in a minute what omics is. And the omics characterization of pediatric acute myeloid leukemia and targeting therapeutics as well. I'm not going to talk to you about my research in chronological order. I'm going to talk to you about it in topics-wise. And I'll go in this order as it is shown. So first of all, few words, general definitions, take you slowly into the subject. What causes cancer? very easy, straightforward answer is gene, environment, lifestyle interaction, meaning that we have some factors in our DNA that will make us vulnerable to cancer, and then with exposure to certain pollutants or whatever you call it in the environment, we can develop, or not we, they, whoever, can develop cancer. So one of the first hypotheses was Knudsen's two-hit hypothesis that shows those two things are the genes. Every one of us has 2 copies of the gene in each cell, one from dad and one from mom, okay? And then Knudsen said that at one point in your life, one of those copies will get mutated. Later on, another copy, the other copy, will get mutated, and only then you will develop cancer, okay? This is for the non-hereditary types of cancers, those that are not inherited. We call sporadic cancer. And then for the hereditary cancer, some of them are, some types of cancer are, we are born with one damaged copy that inherited from mom or dad, and then, we acquire the other mutation through life, and then that's why it's kind of a little bit faster to get the cancer when it is inherited than the other one. Aging is also a factor for cancer because the incidence of cancer rises with aging. Why? Because our DNA repair, we have a repair mechanism. You know, when something goes bad at home, we get the plumber or the electrician. We also, to repair it, we have a repair system in our body as well that repairs DNA damage. So with age, the DNA repair mechanism also becomes less effective with age. I have here an example. Actually, this a little bit weird, this room, because you have to give your back to the audience to show them. So this is an example of what I was just saying about gene-environment interaction. So in the case of liver cancer, I worked a lot on liver cancer, we have certain genes that get mutated. Those are just names of genes. Don't worry about that. It's just names of genes, okay? These are candidate genes that get mutated, and then if the person with these mutated genes get exposed to hepatitis B or aflatoxin, which is a carcinogen in some molds, or smoking, for example, they develop liver cancer. It's not a process of overnight. It takes about 20, 30, 40 years to do that, okay? So cancer is, as I just mentioned, is not overnight, does not develop overnight. It's a multi-step process. So here, this figure shows any human being from birth, his life or her lifespan, from birth till death, natural death. If they have some of those factors in their DNA that will make them more vulnerable to getting cancer, then this is the start. Then those cells will take so many years to transform from pre-malignant, pre-cancerous, until the malignant transformation, where the cells start to invade tissues and spread, and then at the end, death from cancer. So there are several opportunities when cancer researchers can block the initiation and progression of cancer on its pathway from pre-malignancy to deadly stage. This is why early detection and early screening is extremely important. On the other side, I'm going to show you a typical example of how cancer develops in the it's a colorectal cancer. This is a classic example from 1990 that just, regardless of all these names, it's a sequential changes of DNA on the level of mutations and what we call epigenetic changes that follow in a certain sequence from when the cells of the colon was without cancer, then through early stages until the full cancer phenotype. So now, what happens, this is as far as the organs and the lifetime. What happens inside the cell? The cancer cell reprograms, you know, like when you program your computer. So the cancer cell has a computer, it reprograms it in order to acquire certain capabilities and that we call hallmarks of cancer. And these capabilities will allow the cancer cell to resist therapy, not to die, because those capabilities will help or will let the cancer cell not resist any death signal, allow it also to proliferate endlessly, without control, as well as other stuff. I'm not gonna make you get bored of listing all these. But what I just said is that I worked on five of these hallmarks in my life. So here I want to also stress on another important factor in cancer research, which is the model. We cannot conduct research on human beings, of course, unless it is a clinical trial. What we do is that we have to have an experimental system. These experimental systems can be either cancer cell lines that are commercially available, primary cancer cells that we get from the surgery room, from the patient directly, or also we can grow these cells either in what we call two-dimensional on a surface like this, or three-dimensional in a matrix, you know, like a gel. So I worked with all this, or on all these actually, and they're very important because the results you get from one model is different than the other model. The best model you can get, but it's extremely expensive, is this guy, the mouse. So I worked on thousands in my life of genetically modified, I played with their genome and I got genetically modified mice. So this was in America. It's extremely expensive experiment, and but it's very helpful because the mouse is very close to us in terms of the genome, the cells, everything. And the lifespan, I mean the pregnancy of the mice is just 20 days, so it's easy to manage. Also, I worked on rats, on frogs, you name it. Now, the title is called Molecular Pathways and Targeted Therapy. What is a molecular pathway? As you can see here, that's a pathway, is a track where you go from point A to point B on the street or wherever, okay? So the same thing, we have in cancer or in the cell, we have something called pathway as well, like this one. But we describe here how the signal comes from outside the cell. This is the cell membrane here. How the signal, like growth factors, come from outside the cell, into the nucleus to do the function that it's supposed to do, whether it is cell division or cell death or gene expression, whatever, okay? So this is a pathway, and all those names inside the colored boxes are different proteins. So when we say protein signaling cascade or pathway, it's how the signal is moved from outside to inside until the nucleus, like exactly the pathway we take when hiking in the forest. Is this clear? Okay, thank you. The second thing, what is targeted therapy? Because I'll keep repeating this word over and over again. Targeted therapy is like, so you can see on this here that this is a traditional chemotherapy. Traditional chemotherapy is like shooting a grain of salt with a machine gun or a rifle. Imagine, you're just shooting everything around it. Those are the cancer cells, which are in red, and those are the normal cells in blue. The cancer cells, because they have the hallmarks I just explained, they were not gonna die from chemotherapy. Not all of them, but they can acquire certain capabilities that make them come back, what we call recurrent cancer. The poor normal cells don't have those hallmarks, those mechanisms that will make them resist death, and so will die, okay? The second thing is the targeted therapy. It's exactly like playing the darts. We have a dart in our social hub at the School of Applied Science, and this is exactly the same thing. So we have a mutation in the center here, certain mutation or change in proteins, and we hit it with a drug. So this is what we call targeted therapy, okay, clear? So what is omics? The last thing. Sorry if I'm boring you. So omics is a collective name for studying the genome, the proteome, and the transcriptome. We have 3 different molecules in our cells. DNA is the blueprint, gives instructions. RNA is an intermediate stage, and then proteins that I was always interested in are the final product, and on which 96% of drugs are acting. So all DNA in the cell is called genome, all RNA is called transcriptome, all protein is called proteome, and the study itself of the field is genomics, let's skip omics, proteomics. This is why we call omics, because there are tons of omics. There is metabolomics, there is phenotypics, there is connectomics, you name it. So this is why we collectively call omics, okay? And then, don't worry about that. This means that the proteins will just behave differently between normal and cancer cells. So the first topic, finally, of my research is the role of the insulin-like growth factor 1 receptor and its targeting. Now you know what's targeting, right? Hitting it, inhibiting it, and its novel functions. So before I went to Germany, I didn't know this guy. I never met him before. So I was introduced, was introduced in Germany to the insulin growth factor receptor. I'm very proud that I created this, by the way, on PowerPoint. And so I'll bore you with this globe going around and around. Anyway, so in 1995, I traveled from Egypt and went to Germany for my PhD. And it was in the University of Heidelberg, the German Cancer Research Center. I was fully funded by a scholarship from the, a German Academic Exchange Service, or what we call in the short for this word in German is DAAD. Anybody heard of it? Okay. I didn't speak German. Said, okay, who cares? I'm going to learn German. And I had to do the Viva in German after 5 years. So then I was also proud to be the only female in Egypt in 1995 who got a full scholarship. It was a very tough selection process on 2 stages. One stage interview in Egypt, one other one in Germany. And my interviewer, this guy here, Mr. Peitzel, I asked him, he's a professor of Arabic. So I asked him, on which basis did you select me? He said, because I could see that you're stubborn. You're gonna make it. You're not gonna quit. You're not a quitter. So regardless of whatever you're doing there, it's good investment for the money. So I said, okay, I'm gonna do that. So, and these are my colleagues and in 1995, which was a preparation year for us. And then went to Germany. I started working on this phenomena, which is liver cancer. And the status at that time was that we know that when liver cells, normal liver cells, transforms to malignant cells, they pass through different stages. Remember the blue curve here when I said initiation, pre-malignancy, malignancy? So these stages, the earlier ones, involved storage. You can see this reservoir here. Storage of glucose molecules, something we call glycogen, okay, that have red color in the early cells. And then as the cells move towards malignancy or cancer, they start losing this glycogen, okay? So gradually losing the red color until they become totally without glycogen. So we had, I had the rat model of chemically induced liver cancer, and also I established an in vitro cell model in the lab, something synthetic, okay, that mimics what happens inside the rat. And the question, any research in the world has to start with a question. You ask a question, and then you come up with experiments to try to answer the question, and you prove your hypothesis or disprove it, and you can go right and left until you find what you're looking for. So the question was, what is the molecular basis for the transformation from pre-malignant glycogen storing to glycogen-poor cells? What causes this? Is it mutation? Is it an epigenetic change? Is it what? So this is just a very, very little part of the liver cell, showing our friend, the IGF receptor, the insulin receptor, and what's this called? You remember? Molecular what? Pathway, okay? So I had to dissect these, and all these are proteins inside the boxes. Don't worry about the names now. And what we found was that the IGF receptor is a very important molecule in cancer because it controls, it regulates growth, proliferation, motility, invasion. All these are hallmarks of cancer. So we got to do something with this molecule. So we started dissecting, myself and a colleague of mine, Dirk, the different pathways in the liver cell at different time points from pre-malignancy to malignancy to find out what happens to glycogen, why it disappears, why it stores at the beginning and then disappears. And we found out that there are no mutations, so this process doesn't involve any DNA mutations. There are several of these genes, regardless of their name, several of them are changed in their expression, either up or down, and their protein activities as well. And of course, we published all that and we got our PhD. And I continued working on the IGF receptor in Egypt with a purely Egyptian fund, a purely Egyptian scientists. So I got £1 million, Egyptian pound, grant from the, what's it called? Science and Technology Development Fund. And I had master students, PhD students. Because we know from my PhD that liver cancer starts with infection with hepatitis viruses through cirrhosis and then through the malignant phenotype or the cancer, we call it hepatocellular carcinoma. And at the same time, there is very high prevalence of hepatitis virus in Egypt, so I thought, okay, maybe IGF receptor and his family members probably play a role in that. So we, with colleagues from medical school, we got 240 patients, some of them cirrhosis, some of them have the cancer, some of them are healthy individuals. We measured in their sera the levels of several members of the IGF family, and we found that this guy called IGF-BP3 can be used as a marker or a biomarker to predict the development of liver cancer from cirrhosis. Okay, so this was the work of my master student called Asma. And I think here we are all trying to decolonize the curricula, decolonize the research. So that's an example that other people in Egypt can do it as well. Then that was the golden year of grants. I got another grant from the Swedish Research Council on targeting IGF receptor in cancer. And it was in a collaboration with Professor Larsson from Karolinska. And we worked on that. And while working, I got a third grant in the same year. That was really massive. This grant was a big one from the European Union. And I like telling this story. I hit submit 1 minute before the website closed. So I got it, and I traveled to Sweden to work on another topic related to the IGF receptor. What is this topic about? The status of the science at that time said that there was a new phenomena, that IGF receptor, I told you it's a membrane protein. It migrates or translocates or walks or runs, whatever you call it, to go into the nucleus. Something was new at that time and nobody knows why. So the question we asked, what are the biological functions of the IGF receptor in the nucleus? So we know its functions in the plasma membrane. What is it doing inside the nucleus? So that was my PhD student, Ahmed, who was my master student in Egypt, and I got him with me to Karolinska. I helped him get a full PhD scholarship. I want to pass the message, if someone helps you to get a scholarship, please do that with junior ones, with the younger generation. It's a circle, it goes round and round, okay? So I'm not gonna bore you with all these details, but the question, or the observation was that we found that really IGF receptor translocates to the cell nucleus, and the cellular model we used was human embryonic stem cells. So these are not cancer cells. We found that, the question, what's it doing inside the nucleus? So we used proteomics, mass spectrometry, to find the proteins that bind to this receptor inside the nucleus. All these blue color spots are the IGF inside the nucleus with fluorescence, immunofluorescence. Using different techniques, we found indeed that it goes to the nucleus, and by mass spectrometry, we had a list of potential proteins. We selected one of them called the proliferating cell nuclear antigen because it's an important protein. Why is it important? Because it controls or regulates a process called DNA damage tolerance. The DNA damage tolerance is, without details, it's a very important process to maintain genomic stability. genomic stability means it's the opposite of mutations. When I say mutations cause cancer, then genomic stability is the opposite. It's we want to maintain a healthy genome or healthy DNA. So we found, long story short, that, IGF receptor binds to this pCNA and activates it and plays a role in this very important process called DNA damage tolerance, which means that now we are discovering novel, new functions that were never known before. That made it very important. The second question is, what is the function of this IGF in cancer cells? Remember, we were using embryonic stem cells. So now this is done finally at LSBU. Here, I got cell lines with me from America. I have Mavra, who is sitting there in the audience. I cannot, here. And Valentina is co-supervisor, and Chinedu is also a collaborator on the work. And we hope we can finish this and get some real publications out of LSBU, not from our external collaborators. And the second chapter, or the second topic, do you have any questions regarding the first topic? Okay. Second topic is the cell cycle, and from its name is the cell division cycle, okay? That is very important for normal and for cancer cells. Again, took the plane, traveled to America, to the National Cancer Institute in Maryland, and worked with Philip Caldes, again, fully funded postdoctoral fellowship for 5 years, but I stayed only 3 there. And why? Because I was offered a job. Anyway, so why are we studying cell cycle? It's because it's very important, associated with essential processes, with improper imbalance will trigger cancer. So it's very important. How did we? It's again a very good model, a very good example for how important the experimental model is. The status at that time was that the cell division cycle has 4 phases, as you see, colored phases here, red, blue, 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 and pink. And it was thought that protein complexes, like these ones, are very specific for each stage. They cannot exchange functions, okay? They are very specific for this stage. And this was the dogma. So what we did was, we thought, okay, we want to question if this is real or not. So we created genetically modified mice. That was a time that I was working with thousands of mice in the lab. We hit or deleted the gene encoding CDK2. And what we expected based on the previous dogma is that the mice will die, because if it is this important, if the gene is that important, and you knock it out, you remove it out of the cell, the mice should die. But the reality was that the mice didn't die. So if you're wondering what are the positives and negatives, the positives are the mice with intact gene, and the negatives are the one genetically modified that don't have this gene. Okay, they could live, that was a surprise, but they were sterile. So we discovered a new function, that this gene is not important for the regular cell cycle, but for the cell cycle that occurs inside the germ cells, like the ova and the sperm. So the poor mice didn't have ovaries, or have very shrunk ovaries, and also very shrunk testes. And the testes here, the blue color is dead cells. So in the testes, the section of the testes, there were no sperms. That's why they were sterile. So we proved that this is not true, what has been published before. Not only that, we also found the other candidate that compensates for the function of this loss. When this guy is lost, then this guy here can actually run and take all the cycle, control all the cycle, all stages of the cycle. This was a very big discovery, a paradigm shift, and a new model that is taught now in textbooks. And for this, I actually got, it was published in Nature Cell Biology, featured on the cover. I got 3 prizes. I got the Egyptian State Prize. And we were just 3, by the way, who did all this work. Okay? So that was part of my life that I'm very proud of. And now, starting the 3rd chapter, or the last research topic that I'm going to talk to you about, which is, am I good with timing?
Yes,
yeah.
10 minutes.
Okay. And it's about omics. I explained what omics is, and the characterization of pediatric acute myeloid leukemia and targeted therapies. Again, flight from Sweden to Arizona. And working with a renowned late professor, Bob Arcisi, who was a physician oncologist, scientist. And our lab was in this building, very fancy building in Arizona in the desert. We started working on the genomes and the proteomes of the cancer patients, the pediatric, the children cancer, leukemia. Without, I don't know if anybody here has not heard of the Human Genome Project. So that was a very important discovery. All the world was working, 6 countries, 20 centers, thousands of researchers working 24/7 to sequence the human genome. And they worked from 1990 till 2003, 13 years, day and night, in order to sequence the human genome that was published in 2003, the first draft. By the way, at that time, it was only 90% complete, or 92%. The last 8% were completed in March 2022, this year. Okay? So then, why did I mention the genome and the human genome? It's because the information we get out of the human genome for every patient will help us to implement what we call precision medicine. What is precision medicine? Or personalized medicine? Some people call it personalized medicine as well. There are several definitions. I would take the last one. It's one of the goals of this personalized medicine is to use the individual human genome sequences to make accurate diagnosis of a person's risk, developing a disease, and to use the genetic characteristics to plan effective therapies and treatment regimens as well. Because one of the projects I was leading in Arizona was that some children getting a certain drug are resistant to this drug. Why? Which mutations do they have that make them resistant to this drug? Okay? So here it's a little bit simplified. We are talking about only the genome, but it's much broader than that. There is the transcriptome changes, RNA changes, as well as proteome changes as well. So I chose to focus on the proteome changes. Again, I remind you about the darts. By the way, this is the actual dart game in our School of Applied Science. And I was very proud of myself that I got it after 100 times right. So I decided to take the picture and use it. So now look at these patients. Those are those getting the chemotherapy. Those are acute myeloid leukemia patients getting chemotherapy for their disease. All of these patients are treated the same, right? This is what chemotherapy is. But those patients in red, after getting the conventional therapy, the survival rate for acute myeloid leukemia is about 55%. But there is a subset out of these patients that are in gray, their survival rate is just 30%. Who are these and why? Why don't they make it to 55%? These are the ones who have genomic or molecular changes, certain factors, if you will, in their genetic makeup that makes them vulnerable or resistant to chemotherapy. And when they are resistant, of course, they will not be treated and they will die. This is why the survival rate goes down, okay? So how can we know who is going to respond to therapy and who doesn't? I have followed 2 strategies that are not published yet. So I hope to continue on the work here in LSBU. One of them is developing a bone marrow. You know, the leukemia develops in the bone marrow, okay? So one of them was developing a bone marrow microenvironment, mimicking culture for drug testing. And the other one was profiling the proteome of the patients to identify characteristic individual molecular signature for each individual patient. So to develop this experimental model, I want to show you those raspberries and blueberries. Imagine these are cancer cells that are like this, you know, not interacting with each other. On the other hand, we have here the jello containing the same blueberries. This is how it works inside the bone marrow. It's a matrix that have the cancer cell, if you will. Those are the cancer cells, and those are other cells surrounding the cancer cells that secrete stuff that affects the growth of cancer. So we cannot study the blueberries alone and the raspberries alone and the blackberries alone. We have to put them all in the jello, in the bone marrow, in order to get a better idea about the response of the cells to chemotherapy, to whatever targeted therapy, whatever we're using, okay? And we could co-culture the cells together and also use certain fluorescent dyes to identify which are which. Then, this is what I'm trying to do now at LSBU, and this is what got us the collaboration with the industry. We are developing a collaborative project with the industry. Oops. So it's called drug sensitivity and resistance testing, in which we have 384 well plates. Each one of this has one drug, one cell, one type of cancer, okay? We can screen hundreds of these. The device, thanks to LSBU funding, we have this device that could also use barcoding to see the different plates, and you don't have to stay beside the machine. You can go and let it do its job, and then it can give you a readout of which cells have responded to which drug, okay? So we are not getting this one. This alone is like $250,000, so we don't have this device here, but we have another one that could do a similar job, okay? So when you perform or conduct the experiment, you get something, an endpoint like this, 384 wells in one plate. Those, if you follow the colors, some of these are all leukemia cells. These are the different drugs in just one plate. We screened all these. Some of the cells are grown in low oxygen content, similar to the bone marrow inside our body. Some are grown in the atmospheric oxygen to compare which growth condition is better. And then we use the drugs, and those that are red are completely live. They didn't die at all. Those that are blue have died, and we found that the low oxygen content does affect the response of the cells, the leukemia cells, to therapy. Okay, so this is not published yet. We went further to identify, using RNA sequencing, which genes are differentially expressed and are responsible for this process as well. And we found, as you see, those three different columns are like each other, and this one is an exception. So this is the one that gives us clues to why those cells behave differently, okay? So we did the same thing using patient cells as well, and we got some drugs that are working, some drugs are not working, and this is what we're gonna do with our industrial collaborator as well. The last thing was the screening of the proteomic or the proteome, the whole set of the proteins inside different patients with acute myeloid leukemia. And without going into details, we could find that each of this box represent a certain protein that is either highly or differentiated, let's say differentially expressed in different leukemia subtypes, okay? The upper colors are different leukemia subtypes, and this is the expression of those proteins. As you see, there is a certain pattern that could provide a tool to stratify patients based on which protein they express, okay? This is a summary of what we have done before. You have a patient, you get samples from the patient. Some of it goes to molecular profiling on the basis of genome and proteome, and some of it goes to drug testing, and then you get the results. It gives us more understanding of the biology of the disease, the rapid introduction to therapies, and all this will lead to using the scientific discovery for clinical applications, what we call translational cancer research. Translational means you translate what you find on the bench to the bedside, to the clinic. Okay? At one day in my lifetime, or your lifetime, the younger generation, every patient will have their sequence, go to the pharmacy. Here is my sequence, give me the right drug for me. I can see this coming. Maybe not in my lifetime, but in the younger generation's lifetime. Okay, I've talked a lot of science, science, science. How can we translate this to the real world problems? I mentioned some facts about the protein called IGF receptor that could be translated into an early detection tool for liver cancer. And the second one can be used, which is the IGF-BP3, can be used as a predictive biomarker for the progression of liver cancer from hepatitis C infection to whole cancer. We also proposed a new tool or a new assay that can culture the patient cells in a better way, in a more representative way to the physiology, what happens inside the body, so that we can better predict drug response. And lastly, what we do need, all this what I mentioned about omics requires incredible amount of data, we call big data, from the proteins, from the genome, from the transcriptome, from the drug screening, that no one can handle alone. So we use bioinformatics, we use computational tools to make sense of all this data, okay? And we will need to employ artificial intelligence, biomedical knowledge networks to integrate all these data coming from all the processes to make sense. And the challenge will be, how long do we need to do that? There is a cancer patient in the clinic waiting for their diagnosis based on all this data, and waiting to know which drug is better for them, okay? So we need to do this in 1 month. That's too long. Maybe 3 weeks, 2 weeks. Imagine how expensive this would be, but don't look at it this way, because you are not wasting money giving the patient the wrong drug. You are investing time in research, but to get the right drug to the right person at the right time, all the time, okay? See how this will save from the NHS budget. See how this will save lives as well, okay? Maybe there is no cure yet for cancer, but there is incredible progress. These are the 5-year survival rates. Let us say prostate in 2007 till 2013. So, sorry, this was 1970s and this is 2013. It grew from 67% till almost, or 68% till almost 99%. The 5-year survival rate, which means how many patients will survive 5 years after the first diagnosis. The numbers are rising. Look at breast from 74 to 89. Some of them had 90-something%. I don't remember where. Oh, these ones, the thyroid and prostate, almost 98%. So this is progress. This is progress because of cancer research, okay? Now, coming to LSBU, this is the last thing in my life right now. And as I don't need to repeat this because Tara has already included it in her presentation. Maybe what, I don't know if Tara knows that, but we have now a research group. We have Wellequipped lab, we have collaborations with the industry. We recently won an RC1 fund for robotic dispensers as well. And where the future? So I'm trying to implement as much as I can from my experience here at LSBU, applying for grants. We're still working on the functions of the nuclear IGF receptor in cancer. Promising area with faculty with the School of Engineering is the drug delivery using nanoparticles. Also, novel inhibitors from molecular docking with Mohammed Mansour to functional validation, several other things with Valentina and Sinedo as well. Training the next generation biomedical science researchers, moving slowly towards enterprise, and definitely what we need to do to try to do similar work is the human tissue licensing, right? Okay. So I'm very grateful and thankful that I was trained in 3 of the highest ranking cancer research institutes in the world, ranking 3rd, ranking 66, and ranking 99. I thank everybody who contributed to my journey. Of course, on the right-hand side in this yellow box, all my previous colleagues from Germany, from America, from Arizona, from everywhere, from Egypt, all the funding organizations that funded my research, and also LSBU executives and School of Applied Science, Biomedical Science team, everybody who contributed to the Biomedical Science program. I would like to mention two people here, Professor Maria Mavrulido. Why? Because, and she's here with us, because she was the reason why I applied for professorship. I was not planning to apply this year. And then they matched me with a mentor who doesn't know me, who doesn't know who I am. And then I talked to her, and she asked me, so, okay, you're ready to apply? No, I'm applying next year. Why? Because I'm not ready. No, you're ready. No, you have to. Talk to your dean. Do it. And she kept pushing me. And without that, I wouldn't have been a professor today. The second person is Alan, who is also in the audience. Thank you, Alan, who just proposed, suggested. I didn't ask him, because I'm a little bit bad in asking for help. So he came up to me last week. Do we want to rehearse your talk? Oh, yeah, sure. Are you sure? Yes. He gave me 3 hours of his time. Thank you, Alan. It's so kind of you. And thank you. I'm proud to be a cancer researcher. And sorry if anything was boring of what I've said. It's, I think, less than 1 hour. And thank you very much.